Hi, my name is Shannon Oda, and I'm a principal investigator and assistant professor at Seattle Children's Research Institute and University of Washington, and also an affiliate member of the Fred Hutch Cancer Center. And I'm really excited today to present our work in engineering T cells to stimulate endogenous anti-tumor immunity. So here are my disclosures. So it's a very exciting time for us in the immunotherapy field because there's an increasing understanding that the immune system can be harnessed to control and eradicate cancer. This has been shown by several discoveries in the field that have been shown here, both in publications and also by the Nobel Prize that was awarded for the discovery of inhibition of negative immune regulators. So in my lab, we focus on adoptive cell therapy, or ACT, and we like to call this a living drug. And to develop these therapies, what we do is we take immune cells from the blood, typically T cells from our patients, and then we genetically engineer those T cells in the lab to give them genes to have them target tumor. And then we grow millions of those CAR T cells and then infuse those T cells back into the patient. And these living cells can actively migrate to the sites of tumor and destroy the tumors. So there are several advantages to this approach. These T cells can be engineered to be very tumor specific. So it's a very specific approach versus other approaches such as checkpoint blockade. We're avoiding drug peaks and troughs by giving a living drug, and it's a tunable T cell. We can engineer our T cell to turn on and off with different approaches. And what I really want to highlight for today's talk is that these living cells actively migrate to the sites of tumor. So this results in a tumor localized delivery. Engineered T cells have shown clinical efficacy against solid tumors in the clinic. So that's really exciting for us here. I'm showing papers highlighting the success of T cells in pancreatic cancer and also in neuroblastoma. However, we know that there are also several obstacles in the tumor microenvironment that can inhibit the anti-tumor T cell response. We know that T cells compete with tumor cells for key nutrients. There's often a lack of costimulatory ligands in the tumor microenvironment, and instead there's upregulation of inhibitory ligands and soluble factors. T cells can become exhausted, and there's often missing supportive cells within that tumor microenvironment. So in my lab, we've asked how can we armor our T cells to overcome these obstacles? And rather than address each of these issues individually, we're looking for engineering approaches that can address as many of these obstacles simultaneously. So one approach that we've developed in my lab is to develop these novel engineering strategies that use engineered fusion proteins that combine two native proteins to form a new hybrid protein. So what I mean by that is we take the extracellular domain of one protein and combine that with a second protein using the intracellular domain to make a new protein that's not formed in, or is not found in nature, and that we can use to alter the programming of our T cells. So with the extracellular domain, we can choose different domains to serve as a decoy receptor for a negative regulator, for example, for T cells. We can impact the tumor microenvironment, and we can also engage endogenous immune cells. And then with the intracellular component of the fusion protein, we can deliver intrinsic signals just to those T cells that we've engineered to be tumor specific. We can enhance T cell programming and epigenetics, and we can also alter the metabolism of these T cells in studies that we've shown. But importantly, this approach is a low immunogenicity strategy. We're combining two native proteins. These have not been heavily mutated, so they're not going to result in an immune response to our engineered T cells. And again, because we're engineering these living cells that actively migrate to the sites of tumor, we're getting tumor localized delivery. So there are several different costimulatory signals that we can select for the intracellular domain of the fusion protein that can initiate different gene expression programs in T cells to counter several obstacles. So depending on the costimulatory signal we select, we can induce differences in proliferation, enhance tumor cell lysis, alter metabolism, induce resistance to exhaustion, and increase the longevity of our T cells. Whereas most CAR T strategies use one of two costimulatory signals, CD28 or 4-1BB, we can really expand our fusion protein panels to include a variety of different costimulatory domains. So the first class of fusion proteins that I'm going to discuss today is our transformed switch receptors, where we are replacing a brake with an accelerator. So what I mean by that is that we have the ectodomain of an inhibitory or a death receptor that when it binds to the inhibitory protein on the tumor cell, it normally shuts down the T cell. So we've replaced the intracellular domain with the signaling domain of a co-stimulatory receptor, so that instead of an inhibitory signal, these T cells get a stimulatory boost from the transferrin switch receptor, or TSR. And so now they're getting a positive signal from their antigen receptor, a positive signal when the TSR binds to this inhibitory protein, and these T cells are again able to lyse tumor. So one target that we've established for the switch receptor platform is Fas ligand. And Fas ligand has been known to induce T cell death and is a limiting factor for adoptive cell therapy. And this has been shown by multiple papers now. And it's also been shown that it's not only expressed on the tumor cell, but also expressed in the tumor-associated vasculature, on inhibitory cells in the tumor microenvironment, and can also be expressed by T cells themselves as a way to regulate T cells and protect the tumor from immunity. Fas ligand is overexpressed in the majority of cancers. So in this bar chart, we're looking at expression of Fas ligand in tumors relative to the red dots, which is the expression in the normal healthy tissues. And you can see that in the majority of tumors, there is overexpressed Fas ligand in the tumor microenvironment. So a graduate student in my lab, Ben Curtis, did a CRISPR gain-of-function screen in CAR T cells, and he developed a novel vector and guide RNA delivery system to achieve co-delivery of both the guides and the CAR, and achieve sufficient cell numbers such that he could do functional assays with these library CAR T cells. So he then took these edited T cells and then did a stimulation assay where he stimulated with tumor in the absence of supportive cytokines, and at day 7 looked at the expression of these different guides in the resulting CAR T cell population. So what he found with these studies, and this study's been posted on bioRxives now, is that you see these genes that are enriched in the resulting pool of CAR T cells that is associated with positive impacts to the CAR T cells, so they become enriched in the final pool of T cells. But there are also genes that, when overexpressed, result in depletion from the final pool of stimulated CAR T cells. And one of the most highly associated genes associated with depletion from these stimulated pool of CAR T cells was Fas. And it was very strongly significant that if you have Fas signaling in your T cells, you will not see good T cells by the end of this stimulation. So we developed a Fas4-1BB during my postdoc, and the clinical testing for that fusion protein begins this year. So we're very excited for that to advance into clinical trials. But to further advance this platform, we asked, what is the optimal costimulatory domain for these Fas TSRs? And we also did some assay development to vet these fusion protein candidates. And this is work done by Dr. Rima Toomey and Valentina Grillo Alvarado in my lab, and they've been fantastic in contributing to these studies. So we developed a panel of fast transferrin switch receptors, and we were able to get great expression with diverse costimulatory domains. And that when we stimulate these T cells multiple times, what we found is that we are getting a positive signal from that endodomain of these switch receptors, such that we're seeing enhanced enrichment of these T cells relative to our TCR-only controls. As I mentioned before, we're also developing assays to better predict in vivo efficacy. So one of those assays that we've developed uses the IncuCyte instrument, which allows us to monitor T cells killing tumor cells over time with microscopy time-lapse images. So we're able to co-culture T cells and tumor cells. This instrument sits within our cell incubator, and we're able to see these images over time to get a long-term idea of how these T cells and tumor cells are interacting. So with this instrument, we developed the serial killing over time assay, or the SCO assay, where we're assessing the ability of T cells to kill multiple rounds of tumor cells. And this is important because as T cells encounter tumor cells, they can become functionally exhausted and lose their ability to kill tumor cells over time. So this is what the assay looks like with the images from the IncuCyte, where we have red labeled tumor cells co-cultured with uncolored T cells. And so we can track the tumor signal through the IncuCyte technology and assessing how much red signal is in the dish. And you can see here that when we can spike in tumor cells every 24 hours, you see a spike in the tumor signal. And so we add in tumor cells every 24 hours to see how well our T cells can resist exhaustion and repeatedly kill these tumor cells. So this is a video of what the IncuCyte assay looks like with our serial killing assay. This is started at the very end of our first edition, so you can see there's very few red tumor cells left. And then just now we spiked in a second round of tumor cells, and you can see how well the T cells get to work. So they're able to localize to where the tumor cell is. You can see here there's a red tumor cell surrounded by uncolored T cells in the yellow circle. And as the T cells do their job, you can see there's much fewer tumor cells in the dish as they are eradicated by the T cells. So the exported data from these experiments look like this, and you can see where we've added in tumor cells multiple times. And eventually, when we add in this many additions of tumor cells, we see that our control T cells lose the ability to control tumor, and the tumor really takes off and becomes the majority cell population in the dish. So these T cells have become exhausted, and they're no longer able to kill tumor cells. But what was really exciting was to see our fast switch receptor T cells and how well they were able to control tumor cells with successive additions of tumor. And we can see that this is very strongly statistically significant, that we had several switch receptors that were able to control repeated additions of tumor cells. Additionally, we developed assays to use within the hypoxic chamber. There are several tumor microenvironments that have reduced levels of oxygen and thus are less able to support T cell function. So we wanted to ask if our different co-stimulatory domains enabled our T cells to overcome these hypoxic conditions and have a better T cell function under 1.5% oxygen versus 20% oxygen, which is normoxia. So again, we have several candidates that are able to enhance T cell proliferation under hypoxic conditions. And really what we're seeing across these different co-stimulatory domains is a diversity of programming. So we see different levels of activation and inhibitory marker expression, as well as production of functional cytokines, such as TNF and IL-2, which really shows that this is a powerful platform to be able to get a diversity of T cells, and you can select which phenotype of T cells would pair best with the tumor microenvironment that you're targeting. So we tested our panel of fast switch receptors in our in vivo model. So this is our acute myeloid leukemia, or AML model. And what we found is that one switch receptor in particular really showed the ability to dramatically improve persistence of our T cells, much better than the other switch receptors, including the 4-1BB switch receptor that is advancing to the clinic this year. And when we did the survival study, we found that this novel co-stimulatory domain also significantly improved survival, suggesting that there is other co-stimulatory domains that have not been explored within the CAR T cell space that should be looked at more carefully. We next tested this against solid tumors. So this was a pancreatic cancer model, and we found that our novel co-stimulatory domain induced increased polyfunctional cytokine production, much better than the TCR only, and also the fast 4-1BB. And when we tested this in our solid tumor model with a suboptimal T cell dose, so a third of our normal T cell dose, we found that the fast 4-1BB was no longer able to control tumor progression, but our novel co-stimulatory domain was able to significantly suppress tumor progression in this model. And this is pancreatic cancers. This is a very aggressive model to be able to control with T cells. So I want to switch gears now and talk a little bit more about another platform that we're working on. And this is to address the possibility of engaging multiple types of immune cells in the body to eradicate cancer. So there are multiple immune cells that can have different programming, and there are some forms of, say, macrophages or dendritic cells that can be either pro-tumor or anti-tumor. And so how can we engage the right type of immune cells to work together with our T cells to have an anti-tumor response? But we do know that there are several solid tumor obstacles that inhibit anti-tumor immunity, not just inhibit our T cells, but also convert these other immune cells to pro-tumor programming, where they're now promoting progression of tumor instead of eradicating tumor. And one of those immune cell types is pro-tumor macrophages, which pose a formidable barrier to anti-tumor immunity. So here I'm showing an image where you can see there are macrophages forming a physical barrier between the tumor, protecting it from immunity from the patient. These pro-tumor macrophages secrete suppressive factors that impact other immune cells that can block the functionality of our T cells. And these pro-tumor macrophages are associated with significantly worsened outcomes in various tumors. Here I'm showing data from AML, And those patients that have pro-tumor macrophages have a highly significantly worsened outcome than patients that do not have these pro-tumor macrophages. But rather than depleting these macrophages, they can be converted to anti-tumor programming with the right signals. And so what has been shown clinically is that with CD40 signals delivered by CD40 agonists, you can get this conversion of pro-tumor to anti-tumor macrophages, where they're now working together with other immune cells to induce killing and phagocytosis of tumors. These macrophages can recruit CD8 T cells and also present antigen to these T cells, resulting in activation of these T cells. So you can get this coordinated attack against tumors involving multiple cell types. But the CD40 agonist antibodies do have several issues. There are dose-limiting toxicity issues due to the antibody modality. There's inadequate pharmacokinetics and tissue penetration. And this is also a systemic delivery. It's not tumor targeted. So there can be off-tumor toxicity issues with this approach. So we asked if there was a way for us to combine our T-cell therapy with this CD40 agonism to develop a new engineering approach to convert macrophages to anti-tumor programming and create a multicellular anti-tumor immune response. So here we are combining a CD40 ligand ectodomain versus a fast ectodomain. And with the CD40 ligand ectodomain, what we're aiming to do is stimulate endogenous immune cells and alter their programming to be anti-tumor. And we're again combining this with a T-cell co-stimulatory endodomain. So we're supercharging our T-cell programming, and we have a panel of different co-stimulatory domains. So we're again selecting those co-stimulatory domains that will best improve function in our different tumor models. And this is work done in my lab by Simone Gwinnett and Rima Toomey. And so I will be showing their work today. So we first asked if these CD40L dual co-stimulatory receptors, or DCRs, are able to make a better T-cell product. So first pass evaluation of these DCRs, do they actually improve the ability of our T cells to repeatedly eradicate tumor? So we used our serial killing over time assay and found that there were indeed 3 candidates that were able to produce much better T cells that were able to eradicate tumor. And this is compared to our antigen receptor alone T cells, as well as our T cells engineered to express the full-length version of CD40L that has not been modified. So the addition of the intracellular co-stimulatory domain for these T cells significantly improved the ability of these T cells to lyse tumor. We next asked if this would result in increased in vivo efficacy. So we again used our AML model, and we transferred our T cells in with these CD40L DCRs. And one construct in particular showed incredible ability to induce accumulation of our T cells in the in vivo setting. And when we tested this in our survival study in our AML model, we observed a significantly increased survival with our CD40L DCR T cells. And this is a very aggressive AML model. We give the mice tumor, and then we dose with one dose of T cells. So these T cells need to be effective with one dose. There's no IL-2 injection, so they need to be able to survive in vivo without supportive cytokines. And so you can see with our control groups that there were no surviving mice from this study, including the mice, the T cells that were edited with the CD40L full-length version. So it was really only the DCR T cells that were able to promote survival in this aggressive, rapidly progressing model. So we next wanted to test this in the solid tumor model. I discussed a few slides ago our pancreatic cancer model, but I really want to go into why pancreatic cancer is very challenging. And it does feature a hostile tumor microenvironment, and some of the challenges of the tumor microenvironment include pro-tumor macrophages, sparse and immature dendritic cells, and a poor tumor-targeted T cell response. And in this chart on the right, this is showing changes in survival of various tumor types over a 4-decade period. And what you can see is that there has been increases in survival for several different tumor types. But during this period, pancreatic cancer was here at the bottom with very little to no improvement in survival. And the increase in survival in these other tumor types is reflective of increased early detection, increased better therapeutic options, but pancreatic cancer is remains a challenging disease and a disease where we need to have better therapy options. So for pancreatic cancer, we asked if we could use our CD40L DCR T cells to convert tumors from cold to hot. So by cold, I mean pancreatic cancer can have an immune desert phenotype where there's very little immune cell infiltration, or an excluded phenotype where the immune cells are just outside of the tumor but not able to penetrate into that dense tumor mass. And so what we really want to get to with pancreatic cancer and other solid tumors is this inflamed situation where we have infiltration of our immune cells into the tumor microenvironment. So we tested our CD40L DCR T cells in our pancreatic cancer model and found that we do get increased anti-tumor programming of our macrophages. And we can see this in vivo by looking at the phenotype of our macrophages. We see an increase in class 2 expression of our macrophages, as well as a decrease in CD206, which is associated with those pro-tumor macrophages. So we're seeing this conversion of pro to anti-tumor macrophages. We also observed increased anti-tumor immunity from other immune cells. So we looked at dendritic cells and we see increased expression of markers associated with maturation of our dendritic cells, and also, interestingly, an increase in antigen-presenting B cells, which is something that we're very interested in following up, and how these B cells could be contributing to an anti-tumor immune response. The next experiment we did was a challenge study where we dosed our mice with a suboptimal dose. So this was 10% of our normal T cell dose. And what was really exciting to see was that even at this low T cell dose, we are seeing therapeutic efficacy with our CD40L DCR T cells, that they're significantly controlling tumor when our control T cells are becoming exhausted and losing the ability to control tumor, which is similar to what we saw with our serial killing assay in vitro with the IncuCyte instrument. So very nice correlation of our in vitro results with our in vivo results. So with our DCR technologies, we are aiming to reprogram inhibitory myeloid cells to induce anti-tumor function. We are observing this shift in programming from a pro-tumor to anti-tumor programming in macrophages. And with our dendritic cells, we're seeing maturation, which can then lead to a more diverse T cell response so that we can go after a variety of targets with our tumors, not just the ones that our T cells have been engineered to target. We've observed significantly improved outcomes in both hematological and solid tumor models, and we also observed clinical efficacy or in vivo efficacy at a suboptimal dose. So in conclusion, we've developed these fusion protein technologies where we have a flexible platform where we can use different combinations of ectodomains and endodomains to produce a variety of enhanced T cell products, as well as start to address some of those inhibitory obstacles within the tumor microenvironment, such as those inhibitory immune cells that can be converted to instead synergize with our T cells to have a significantly enhanced therapeutic efficacy. So with that, I'd like to thank the members of my lab, as well as former members of my lab that have contributed to the studies that I've presented today. I would also like to thank our collaborators and our sources of funding, and very much looking forward to future discussions of how we can continue to develop therapies for cancer that are more effective and less toxic and can have a long-lasting impact for our patients. Thank you for your time.